Hello, everyone. My name is Lauren Griffin, and these are my partners, Angela Dagnino and Ali Fischer. Today, we are going to present to you our research on the directed evolution of selenite resistance. This research was conducted under the guidance of Dr. Noel Reynolds. Selenocysteine is a 21st genetically encoded amino acid and can be found in all three domains of life. Selenium is an essential micronutrient, but is toxic to cells at even a relatively small amount. The mechanism of toxicity is through the misincorporation of selenocysteine at cysteine codons due to the inability of the cysteonyl tRNA synthetase to adequately discriminate between the cysteine and selenocysteine amino acids. We work to perform directed evolution experiments in E. coli for increased resistance to selenite.
To provide some background on selenite, its accumulation in cells as a result of misincorporation into translated proteins is responsible for its toxicity. At the UGA codon, selenocysteine is inserted using a special recognition sequence in the messenger RNA. When the selenium anion, selenite, is taken up by the cell, it is reduced to selenide and enters the cysteine biosynthetic pathway, subsequently producing selenocysteine. While selenocysteine is essential in the cell, it is also toxic due to misincorporation at cysteine codons. This misincorporation is a result of cis-RS's or cysteonyl tRNA synthetase's inability to differentiate between cysteine and selenocysteine during transcription. There is a selenium accumulating plant, Astragalus bisulcatus, that contains a cis-RS variant H235N active site, which has been shown to reduce such misincorporation of selenocysteine at the cysteine codons, lowering the toxic effects of selenite. Insertion of the amino acid variant into the E. coli cis-RS's active site has been shown to lower the toxic effects of selenite as well. Recent studies by UIS alumni students showed that the directed evolution of cis-RS can increase a cell's resistance to selenite up to 5 millimolar. To gain insight into the specificity of cis-RS substrate binding and to determine if we can further increase resistance to selenite, we perform directed evolution experiments up to 8 millimolars of selenite.
Taking a look at this first figure here, this is the selenocysteine metabolic pathway. Starting at the top, you can see selenite being reduced to H2SE negative. Then it enters the biosynthesis pathway, which results in the formation of free selenocysteine. This free selenocysteine is then misincorporated into proteins at cis codons. To do this experiment, we used a copy of the E. coli cis-RS gene that was placed on a plasmid. This plasmid was transformed into a strain of E. coli that contained a temperature-sensitive version of cis-RS encoded in its chromosome. So at 42 degrees, the chromosomal form of cis-RS is inactive and the cell relies on the plasmid-encoded version. We performed a serial transfer experiment, increasing the concentration of sodium selenite every 3 days for a total of 69 days, up to 8 millimolars of selenite. The cultures were transferred every 24 hours and shaken at 42 degrees Celsius. We evolved 3 independent populations of both E. coli containing wild-type cis-RS and containing cis-RS with the H235N variant. After the 69 days of serial transfer, plasmids were removed from the cells and transformed back to a fresh culture of E. coli. These strains were grown in 96 well plates containing 150 microliters of culture containing selenite and were incubated overnight at 42 degrees without shaking.
So to explain the graphs showing the evolved populations showing increased resistance to selenite, the first graph on top, graph A, shows the growth of evolved populations containing the wild-type cis-S, while the bottom, graph B, shows the growth of the evolved cis-S H235N variant, an increase in concentrations of sodium selenite. The strains were grown for 24 hours at 42 degrees Celsius, all in 96 well plates, as mentioned earlier. As shown in graph A, the resistance was highest in the C strain population of cis-S compared to the other unevolved strains. Moreover, populations A and B showed a growth effect as the concentration of sodium selenite increased. The second graph differs from the first in that all the populations of the variant showed significant resistance to selenite even with the increasing concentrations.
Overall, our research showed that one evolved wild-type cis-S population shows significant resistance to selenite compared to the unevolved strains. The other two populations showed only limited resistance to sodium selenite. However, all of the evolved cis-RH S235N showed significant resistance to selenite compared to previously published data on the H235N variant. We predict that the increased resistance seen is a result of novel mutation in the cis-RS gene. In the remaining time on this project, we hope to sequence the plasmids from the populations to determine if this is indeed the case. We hope future student researchers can continue to research the directed evolution of selenite and provide further evidence of our conclusions, as well as gain more information to apply their knowledge in hopes of reducing selenite toxicity in the soil. We appreciate all of your time and are looking forward to hearing your questions at the Q&A.